Right. So we're going to resume the session for next generation sequencing. So as we have the no schedule for tomorrow class, which is the Wednesday one. So I just tried to make sort of replacement video for your lecture. So we can go from the next generation sequencing part, which we stopped at the Illumina sequencing last time. So we continue to discuss from this part. Okay, so as I explained yesterday, Illumina sequencing introduced in next generation sequencing market as Solexa platform in 2006. So first, the sequencing platform was developed for Solexa system. So this machine also based on the method of sequencing by synthesis. So that's also adding the nucleotides for templates and add up the sequence to make an entire sequence information for the read. And another feature is this makes you the immobilized and amplify single molecule on a single surface. So so if you have like tiny the panel things, the solid panel things, then you put on the primer station onto here, like these tiny things, and then you attach the sequence library into the primer station. Then you make the sequencing process. So this is different situation than pyro sequencing. So pyro sequencing use the well system. So put the base, put it on top well, and you do the sequencing. But the Illumina sequencing, you basically make a sequencing on the surface platform, right? And this solid surface, we specifically called it flow cell. So flow cell is something important. So if you go to the link on YouTube, and you can see how this the flow cells is working for the system. So block it. Okay, so this is the video made by Illumina, and you have this the solid panel, right? So solid surface thing. So if you look at this panel closely, then you will see tiny dots on here, right? So this is a little bit lower resolution, but it actually has the little tiny hole like in here and also here. So you basically put the liquid on here and go through this stream and out to this position. So this is basically make a flow from here to here. Then you basically put the reagent and the DNA library mixed into the liquid format. Then you put that liquid on here and put it out to this flow. And onto this flow cell, if you look at closely, okay, so this is the hole I mentioned previously. So you look at here, then you see tiny holes in here, and you basically flow the DNAs and so libraries and also reagents as well. Then if you look at the flow cell closely, then it also has a bit of the tiny primer station, like in this. Then what you have, okay, so within this primer station, then you have the individual positions that can have the library onto this the station area, right? So this station area looks like this, right? So these are not the DNA library; these are the individual station within the entire position. So that has the some primers onto here. So primers means that the artificial DNA sequence templates that can bind to your DNA adapters, right? Then this is the library with the adapters, right? So this is the DNA, right? And these are the adapters, right? So adapters can do several functions. So if you use these adapters, it can use as a the position where primer is going to bind, right? So primer is bound to here, then you can do the PCR to amplify your DNA sequence, DNA libraries. And also these adapters can binds to the individual primer station, right? So individual primer station are these, right? So if you look at library structure, then, you know, I mean, I highlight again, these are your DNA from your samples, and these are the artificial adapters. So this can be like around 100 base pair artificial sequence made by company. And these DNA sequence are all random things from your samples, right? And you put all of the reagents and the DNA libraries into the liquid, then put it into flow cell. Then each libraries will combine around these primer stations, right? And using the reagents, and these are forming the environments that can, you know, the library can be bound to the primer station. Then once it's bound to the primer station, then it will start the DNA sequencing. So if you see here, so from the another strand of the primers, then it start of the synthesis, right? So synthesis means that based on these templates, so template here is your DNA library, right? DNA sequence. Then if that is like, let's say this is the A, then your primer is going to bind to T because this is the complementary nucleotide, right? Then if the next sequence is the probably C, then it will bind to G, right? Then you have the individual, the synthesis for the positions, right? And then after this, and once all of the syntheses are done, then it will turn around the position. And also this is the, you know, conducted by the reagents you put it into the flow cell, right? So these reagents makes the flip around that the library. So it basically flip that position like in here, right? So it will bind to the different, the ends of the adapters, then the bounds to the purple primer station, right? And then once it's bound to the next position, then also it start the sequencing again, right? So sequencing like the first position. Then once it's all done, then you will see whether these positions have the certain amount of the fluorescence or not. So one thing I missed in here. So once you have all of these sort of primer stations, back to here. So it has lots of the sort of the arena where primers can be, you know, bound to the DNA library, right? So in theory, you put one DNA library into this area, right? So how that make happen? You basically make a exact concentration to put it into your flow set. So if you put too much, then there are some possibility where two libraries bound to this area. So you have to make a sort of accurate adjustment by, you know, calculating the molarity. Then you put the one DNA library into this area. Then once it's getting to there, then it will start the amplification around these positions, right? So why that is the important things is once you get the sequencing, then you will see, okay, this area all composite by purple color, and here is the red, and here is blue, and here is green. The reason is that because the one DNA, the library put it into here, then it will bind to all of the area and occupy the entire arena for this space, right? And also that happened to the other positions as well. So once you get the synthesis, each synthesis happen at the one cycle. So in a single cycle, that will give you which nucleotides put it into the which positions, right? So then you can read out that, okay, so probably from here, this will be T, this will be G, and this will be A or something. So how many? That happens in a flow cell, it has the millions and billions, right? So lots of them, then you can read out the DNA sequence information out of this flow cell. Okay, so so back to here. So Illumina sequencing used the reversible terminator sequencing with 4 color dyed labeled nucleotides, right? So we'll come back to in here. So once you get the primers on here, then you get the primer position on here, then you do the sequencing like this way. So okay, so video has this position, this direction, but the scheme on this review has this direction. This is probably due to the years of the review I used in this the video. So this one, I think this one is much earlier version than the video one. This is probably from the 13 or something. But anyway, the basic the principles are same. So once you have the primers onto the library, then you get the each position sequencing, and you get the synthesis by the sequencing, right? So the first position, the first cycle has the certain nucleotides on this and this and this, right? Then once the first cycle is done, then you get the fluorescence emit, and then after that, you basically cleave the area of the fluorescence. Then it will make the hydroxyl groups on here. Then it will bind to the another the nucleotides onto here. Then it will repeat the same synthesis from this process. So the basic idea is that you get the multiple cycles to synthesize your sequence, and this cycle means that how many times you can read out for your length, right? So let's say you have the 13th cycle, then you get the 13 base pair readout, right? If you have the 100 cycles, then you have the 100 nucleotides readout. So each cycle you can read out the single nucleotides from that. Okay, so during each cycle of the process, the sequencer wash out the chemicals that include the variants for all 4 nucleotides over the flow cell. So that's basically the summarize what I said before. Then the nucleotides have blocker, terminator, cap, so that one base pair can add it to each molecule of DNA at a time. So in here, each nucleotide has the fluorescent, but also blockade as well. So there is no way another nucleotides can be bound onto the next position, right? So each cycle has the only one nucleotide added. Okay, so after the detection of the fluorescent signal, the blocker can be removed, then cycle can start again, right? So this is the happen at the cleavage, right? And then after that, you get the entire sequence information from this synthesis of the sequencing, right? Okay, so I think that is basically the same idea as before. And then, right, yeah, this is the summarization. Okay, so once you added the cycles onto each flow cell, then it will be detected by the camera based on this fluorescent information. So once you get the image at a certain cycle, so let's say the first cycle, then you will see each position's nucleotide information based on the fluorescent, right? So you can read out whether that is the A, C, T, G by the colors, right? So if you have 6 cycles, which means that first, second, and the third, fourth, and sixth. So you repeat the cycles, then you will see the information for each cycle: first, second, third, fourth, fifth, and sixth, right? And you can read out the DNA sequence information. So let's say on here, this is the one library strand bound onto the primer station, like in here, right? That's probably this one, right? And then this is the first cycle. So first cycle is C, so green color. And the second cycle will be the same position because it will camera onto the from the top, right? And the second cycle will be A. So that's not. So this is actually the first cycle is C, and the next one is the A, right? So it's not picturized on here. So this is a C, A, and the third cycle, the nucleotide is T. And the fourth one is C, and G, and what is that? The T, right? So the DNA sequence of this strand is C, A, T, C, G, T, right? But if you look at this strand, so this is must be one of these, right? And there would be G, T, 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 C, T, right? So you can read out the 6 cycles out of that. So Illumina sequencing is quite accurate in terms of reading out the individual sequence, and also why Illumina sequencing sort of dominate the market. The reason is that they can read out the large amount of DNA library read from their sequencing machine. So the good thing is if you look at their machine, so if you use the Illumina HiSeq, this is like the iPhone XR, iPhone X, or whatever, iPhone 12 or something. So there's a lineup for the Illumina sequencing machine. So Illumina HiSeq is one of the large machine at early days. So I mean, this is the first version of sort of like monster machine. Illumina HiSeq, they can produce 3 billion paired 100 base pair reads. So they use the 100 base pair reads. So 100 base pair means that they can read out the 100 cycles of that. So they can read out this process. They repeat the 100 cycles, so they can read out the 100 base pair length. But as a sum, they can read out 3 billion paired sequencing from their run, right? So if you want to read out the 600 gigabytes data set, then you can get the like $10,000, and it takes 8 days, right? So each day takes around 70 gigabytes something. So 70 gigabytes is way higher number than we used in the Senya sequencing, right? So that takes only 40,000 base pairs, so which is way less than 50 gigabytes one, right? So where am I? And here, Illumina 10x. This is another version of the Illumina HiSeq. So this is larger than HiSeq, so that can read much longer read and also much more data set. And more recent version is Illumina Nova 6. So that is the sort of the largest the output of the sequencing. The Illumina also make sort of the cute things like Illumina NextSeq. So it takes I think it produced much smaller version than this. But why people want to use this? Because this is so quick, right? So these machines are large, so you have to wait for the the samples. But sometimes you want to screen, you want to get the sequencing data for only one person or only one virus genome. That case, you use the Illumina NextSeq. So this machine especially widely used in these days for COVID-19 sequencing, right? Okay, so good thing for Illumina sequencing, it has the best throughput. So you have the more data set that will give you more confidence. And also you can sequence multiple samples that will win out for the research and also the clinical use as well. And it has the accuracy and also read length for the good read length for the NGS, but not really long, but it's okay. And also the very fast and robust library preparation. So fast is easy, so it's easy to make up the library. So what is the robust? Especially for Korean students, I never get to the point of the robust during my the graduate school. Robust means that anyone can do this, right? So library preparation is of course quite tricky. It could be tricky at the first time, but once you get 2 or 3 times, then you can do this. You can do this, right? So robust means that everyone can reproduce, everyone can repeat your work, right? So robust means that it's reliable and it's trusty, and also it's straightforward to use, right? So robust end up meaning. So, I mean, compared to other library preparations, so for instance, like, chiral sequencing or other sequencing machine, Illumina is way robust than other platform. But there are some few disadvantages. So very well-known disadvantage for Illumina sequencing is they have the shorter read length. So they have only 100 or 150 base pair for their length, right? So and also the problem is that they are based on the sequencing by synthesis, and they use the cycle concept. So once you get once the cycles goes on, then the quality of the sequencing getting down, right? So this is the examples of the Illumina sequencing. It has the 300 cycles, right? So that means that you read out the 300 base pairs, 300 nucleotides, right? So if you look at the first, second, third, fourth, fifth, and sixth, this y-axis is the quality scores, right? Your quality scores is quite high. So these quality scores you call the Fred scale score, which means that this is the minus log 10 value. So if you have the quality scores, let's say this is 38, but let's assume that this is 40. So 40 means that this is minus log 10 value. So it has to be 10 to minus 4. So what that means, so they meaning the probability of getting error in your readout, in your sequencing, right? So the error rate is 10 to minus 4. So if you do the 10,000 times for, it's a 1 in 10,000. So 1 in 10,000, you will have the error in your sequencing. But if you look at other sequencing platforms like, Ion Torrent or the nanopore sequencing, which I will tell you later, that will has the quality scores around 20. So this is quite high differences between the Illumina platform and others, right? So if you look at the earlier stage, that has the quite high quality scores, but the quality of the sequencing getting down, especially after, about 70% of the cycles. The reason is that if you remind the sequencing process of the Illumina over here, they basically use the enzymes to synthesis and also cleavage as well, right? So enzyme is always do the degradation over the time by temperature or a certain situation, right? So after a few cycles, right? So each cycle, I think it takes like 30 or 40 minutes. So after a few cycles, it would probably pass a few hours. Then after a few hours, the enzymes get degraded, and it has the less accurate, so it will introduce the error rate. So the sequencing has some quality range from the 20 to 30 something, right? So cycle is something problem in an Illumina sequencing. So that's why the quality gets down. And little TMI, this is not that important, but some students might be curious about why the quality score is not highest on the first few cycles compared to like this. The reason is that this process, especially for the first 2 to 5 cycles, we called it priming stage. So what is the priming? So priming is that makes the baseline out of the fluorescent information. So the first few positions, like in here, so let's say this is the first, second, and third, and fourth position, then this Illumina machine take a image out of this information, then it will identify which position has the coordinate out of this. So Illumina adjusts their positioning and also the image process at the first few cycles. So that's why it takes some, the less quality because of this priming and because of this adjustment time, right? So once the adjustment is done, then it will start the highest quality of sequencing throughout few cycles, and after that it will be degraded, right? Okay, so that is the sort of basic backbones of the Illumina sequencing. Then also another core concept of the sequencing, we do the single end and paired end sequencing. So single end paired end means the single end is literally you get the sequence information from the one end, right? So you have the library like in here, right? Okay, so this is the sequence library, right? So library is again, once you get the your sample's DNA, then you make the fragmentation first. So you make the fragmented into the 30 base pair something, 30 to 500 base pair. Then you attach the adapters by ligation to each end of the DNA, right? Then you denature this double strand library into the single strand. Then you put this library into the flow cells, right? So this is the same concept you see in here, right? Okay, so once you get the library like in here, then you can have the two options. So first one is you read out the information for one end, or you can get the information from the opposite end, right? So if you get the only one end, then what means that it means that the single end, right? But paired end is that you get the information for both, right? So again, this is your library. Then you get the only one read. So one readout for here. So one readout means that you get the information on this area right here, right? But if you have the both adapters here, so you can basically turn around the library, then you sequence for the opposite direction. Okay, now I'm here. Okay, so you basically get from your library, you get the two reads, first read and the second read. So if you have only one, then you call single end. If you have the both end, then you call the paired end, right? So what are the good things for the single end, paired end? So single end is obviously cheaper than paired end, right? Because it's only the one way. So it has the, it's less than a half price for the sequencing because it only gives you 50% information. Paired end gives you the extra cost, so it's double up the cost for sequencing. But good thing is you can know about the position of your reads more accurately. So if you have like you get the DNA sequence from the repeat sequence, then the paired end sequencing has some information about the the positions as well, because this position and this position they're having the information for the library size, then it will exactly read out the positions for the sequence information. Okay, so paired end sequencing. Yeah, reference genome. Okay, so that's all good. So more specifically, so if you look at the paired end sequencing, then you have the first read and second read, right? So you can take advantage of this paired read. So let's say if you have the deletion on this position, then your read is going to bounce to more shorter than before, shorter than reference, right? Then the reason is that your samples has the deletion on this position, then your read between these two, you have the much shorter distance than the reference genome. Then you can notice that, okay, there must be deletion in this position, right? Or if you have longer paired end distance between these two reads, then you can estimate that there must be extended things happen in this position. So that means that DNA duplication mutation happen on here. So that's why you have the much longer distance between these two, right? So this paired end sequencing has much more accurate information. So I think in these days, most people use the paired end sequencing. So I mean, probably there is no point we can really thought about the differences between the single and paired end. But what I want to emphasize that the paired end information has the sort of specific advantage of why we have to use paired end in the DNA variant discovery or in your genomic sequencing. Okay, so next section is for the next generation sequencing of the long read sequencing, right? So long read sequencing literally means the longer read, right? So why long read sequencing introduced into the field, especially after the Illumina sequencing platform? The reason is that Illumina sequencing has the shorter reads, so people are not really happy with that platform. So people try to make a longer reads, then we can utilize that information into the genomic research, right? So one first frontiers in this field is the PacBio sequencing, and they also use the well system as others, right? But this time they can modify their polymerase, then they can read much longer sequence than before. So yeah, most of the ideas are quite same. I don't know what to. Yeah, basically sequencing are same, but they natively use much longer read library preparation in their templates. So like in here, Smart Bell template. So they can use the 2 hairpin adapters. So rather than they put it into the single strand library, so they put much more stable format into the sequencing situation. Then they can read out that sequence information, by fluorescence as others used in their system. So the point is that they obviously can do the longer sequencing. So let's say Illumina is like 150 base pair, but this is a PacBio sequencing that it will produce much larger library readout. So one reads can take up like 71 kilobase, right? But it has like a 10 kilobase things for their sequencing. But the problem is that it has the high error rates. So it has around 1 or 2%. So let's assume that 1%. So 1% means that 1 in 100, right? So 1 in 100, if you take a minus log 10, then it will be 20. Minus log 10 times 10, right? So it has to be minus log 10 multiply 10, right? So minus log 10 of 1%, that will be the 2, right? And you have to times 10, right? Then it became 20. That is the Fred scale. So if you look at the Fred scale here, then you have the 30. That means that 1 in 1,000 error rate. So 1,000 base pair, you get the 1 base pair error. So that is the 0.1% error rate, right? So if you take the minus log 10 times 10 scale, which is the Fred scale, then it will be the 30, right? Okay, so, and also it has the lower throughput. So if you run the one sequencing machine, then you get the run 5 gigabase per run, right? So if you think about the differences to the Illumina sequencing for the one run, it will produce the 600 gigabytes. So it's about 1 in 100 throughput. So lower throughput means that it will be more expensive than the Illumina. So if you want to sequence the one person's genome, so if you want to sequence your DNA genome, then you go to the company, you ask, I want to do Illumina sequencing, then the company asks you, okay, so let's pay $2,000, so which is about 2 million wons, right? So it's okay. So it's about laptop price, right? But if you ask PacBio sequencing for the run, it will take $30,000, so which is about 30 million wons, right? So it's 10 times expensive than Illumina sequencing. So that's why PacBio is still, it's definitely good technology, but takes some time for the general use. Okay, so PacBio sequencing, what is good for? It's good for the genome assembly. So think about the situation when we assemble the puzzles for the reference genome, then shorter read has the problem. So people want to use the longer read for their assembly, right? So PacBio sequencing has ruled out that information. And another technology for the long read sequencing is the ion torrent. I'll little bit hesitate. Ion torrent is the long read sequencing. It could be somewhere between short and long. It's longer than Illumina, but it's not that long. So, but anyway, it's definitely longer than Illumina. So, but anyway, so ion torrent is literally means using ions. So ion torrent is a bit different from the PacBio or other Illumina sequencing platforms because it uses the ions rather than fluorescent, right? So once you add up the sequence like in here, so it's also based on the sequencing by synthesis, right? It's emit the ions rather than other than the fluorescent, right? So it emits the hydrogen, right? So when in one position and another position, it emits the ions for each cycle. So each nucleotide has the different levels of ion emission. So A has, let's say, this degree, and the T has the longer degree. Then it will notify that which sequence is composite in these positions, right? So Illumina sequencing, ion trends measure the current from the ion emission, and it specifically process the position on the semiconductor platforms, right? So it modified the semiconductor positions to sensitive onto the ion detection, and using this ion detection with current level, it will identify which sequence binds to each template. So ion trend is good, but it has some problems into the repeat sequence, especially if you have the sequence like an or T or A, then your current level is a bit less accurate in terms of detection. So ion trend is, because of these issues, and people not really wants to use the technology, and also the popularity is getting down, right? And also another problems compared to Illumina sequencing, this is totally different aspect. This is more like a computational science. So once the things has to develop for the computational process, then the platform has to be discussed in an open platform, right? So Illumina sequencing, once the data generated, people develop the algorithm, people develop the scripts in the open space. So they open up their script and they open up their algorithm principles and people discuss. But ion trend, my opinion, ion trend is failed in that sense. So they're very closed, and they provide their algorithm throughout the company. So without the commercial license from the company, then we cannot adjust their the analytic framework. So that is one way why people not choose ion trend for the development, right? So this is like the situation for Android and iOS. Of course, iOS is much closed format than Android, but in early days, both platforms has really open for development. So people want to develop their application and also get some rewards or the advancement out of that environment. But in the end, iOS from Apple or some the platforms, they close their development the word, and it became less attractive for the people. But iOS, it takes a little bit different strategy, but back up there, it's quite similar situation. Okay, so another longer sequencing technology is the Oxford nanopore technology, and this is also used the hairpin templates, also makes much stable information. Then during the sequencing, it unwinds the hairpin structure and put it into the pore structure of the sequencing process. So once the DNA sequence unwinds to this pore structure, and this pore takes out the DNA sequence into the hole, and within this, the whole structure will measure the currents out of the DNA sequence. Then it also reads out the information based on the current, and it will identify which sequences belong to this template. And this is again quite similar concept as ion trend, but the good thing is the molecular mechanisms of this pore structure is much stable than ion torrent, and also Oxford Nanopore has much longer and much stable format in their library preparation, so using the hairpin template compared to the ion torrent platform. So based on the beads and the single strand format, so Oxford Nanopore developed in the much more stable format, and it can read out the much longer information than ion torrent, right? So Nanopore sequencing also has the longer read than other platforms, but not much as the PacBio, but it has quite longer, like 4 to 6 kilobase, and I think this is much longer these days. So if you go to Twitter, Nanopore users always show off their longest read of their life, right? So you can read out how much longer they can go on these days. But anyway, so Nanopore has the longer reads, and also it has the it has very portable format, right? So this is the machine of the Nanopore. So this is like it's a little bit bigger than USB. So this is like probably 1.5 times bigger than USB, but it's quite small. So this is like an MP3 players. Okay, so this takes really MP3 player size, right? And this is tiny. This is cute, portable. So you can bring this machine to anywhere you want, right? So when Ebola happened in Africa a few years ago, there was really concerning situation at the time. So people from the MIT, Boston, they bring their nanopore sequencing machine, then they visit the Africa bravely, and they sequence the Ebola virus sequence from the Africa, right? This is amazing. This is really kind of imagined by PacBio or Illumina sequencing machine. So this is the Illumina sequencing machine, and this is like a fridge size. So this is, I mean, this is bigger than me, and this is obviously way bigger than me. And this next thing is like, you know, probably desktop size. So this is also quite big to bring, right? And also ion torrent is big as well. PacBio is quite large, so quite chunky, so kind of bring it. But you know, Nanopore is tiny one, so you can put it on your pocket, bring it, and to anywhere you want. So you can basically read out the information. And okay, so this is the size. Then you can connect to your laptop. Then you can sequence it, right? And also you can bring the Nanopore to the space. So this is in the somewhere in a space station. So I don't know. I mean, I can bring the Nanopore to the space station, but I don't know how to get to the space station. But anyway, so if you want to, if you have planned to visit Moon or Mars with the stupid guy Elon Musk, then you can bring your Nanopore to that place. But yeah, okay. So and also some virus people they use the Nanopore for the for other search of the virus sequence as well. And you have the longer sequence, then obviously you can read out the large genomic change more specifically. So if you use the Illumina sequencing, if you read out the some 100 base pair deletion on the genome, this is quite messy. So this is the reference sequence, and this is the each read. So this is about 100 or 150 base pair reads, right? And you get the missing position here because the homozygous deletion happened at this position. But if you look at the Nanopore, Oxford Nanopore ONT, then you can specifically measure the types of deletion, right? So if you measure this position by Illumina, the Illumina gave you homozygous deletion onto this position. The reason is that the reads information totally missing in this position, right? So if you look at here, if you stack up the reads, if you pile up the reads in a specific locus, then you will see the depth. So let's say here is probably 50 or 70, because this is a 76, I can see that. So it's only seen by good person. So there will be the max position is 76, so 0 to 76. So this is the depth in each position, right? So 76 was 60 something, 50 something. But anyway, it's between the 0 and 76. But this part is totally 0, right? So you can say here is probably homozygous deletion, right? But if you look at ONT, then that will call you the heterozygous deletion. The reason is that only about half deletion happening here, right? Oh, yeah. Half deletion, half depth are reduced, right? So if you think about the 2 strands of the chromosomes, then you have the half position on here, half position here, right? So if you have the heterozygous, then you have the half reduction of your read depth from the sequencing. Okay, so the point is that the longer read sequencing gives you much accurate information for the large genomic events like structural variation or copy number variation. Okay, so next point is the base calling, which is the process to find sequence from the sequencing data set. So any sequencing machine gives you the information for your nucleotides, right? So the first information you get from the sequencing machine is some signals, right? So the signals can be based on fluorescent, or it can be based on the current, based on the ion emission or something. So all of these signal information has to convert into the certain nucleotide positions, right? So this process we call the base calling process, right? So if you have the Sanger sequencing, if you have the PacBio, Illumina, ABI system, this is 454, and this is the nanopore, then it has their own signals, their own information, whatever is fluorescent or current, and it has to be converted into the nucleotides, right? So let's say if you read out the raw signals from the 454, if you read out the tens of the positions, then you get some raw signals from the 454 system, then use their own base calling algorithm, and then you convert that raw signals into the DNA sequence, ACC, blah, 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 blah. And this DNA sequence, we don't know their position, right? We don't know their position. That's the important things, but we're going to discuss in the later part. But base calling algorithm means that they convert the signals into the DNA sequence. And these algorithms is very specifically and very patent in each company. So we don't know the exact details on that, but it takes some algorithm, then convert the raw information into certain sequence information. So what I want to show you is that if you look at someone's genome and you do the sequencing, right? So let's assume that you are a doctor, or you are a some person screening the DNA information. Then first thing is you collect the blood samples from the person, right? So you collect the 5 milliliter of the blood. Then if you look at the blood samples, you have the lots of cells into the blood, right? Within the blood, right? Then lots of cells means that each cell has the DNAs, right? It's the nuclear genomes, right? And you collect the DNAs from the blood. So that process we call the extraction. So you extract the DNA from the blood, right? So you extract the DNA from the 5 ml blood. That will be like, few tens of the micrograms of DNAs, right? Then once you get the DNA, then you bounce that DNA into the Illumina sequencing machine. So once you transform to the library, then you put it onto the DNA sequence information. Then this person has quite interesting, because if you look at some locus, this position, it has this is the reference genome, and this is person's genome, right? So we are human, so we are diploid. So we have 2 strands, but reference genome takes only the 1 strand out of that. So 2 strand, and if you look at 2 strand, it has the TT, right? So this is the same T as reference. So how we call it? We called it homozygous for the reference allele. Or shortly, we call the homozygous reference allele, right? Because it's similar to reference sequence. And if you look at the raw data set, which is from this base calling algorithm, so this is the DNA sequence from the machine, right? So this sequence information is stored as a certain file format called FASTA, right? This is not pasta, this is FASTA, right? So the FASTA format looks like this, right? So 2 information, it's a four sequence information. 1, 2, 3, and 4, right? So this is the ID, this is the ID, and this is the sequence, right? And these alien characters, these are the qualities, right? You cannot read this quality information. I mean, if you're a machine, then you can read it, but I mean, we are humans, so we cannot read it. So this is human readable, right? So ACC, TCG, AAC, which are the sequence of this, right? And these are the same as on here, right? And this sequence is different. No, this is the complement of this sequence, right? So this has to be in an opposite position. So we have the 2-to ratio, but I mean, we might not have the 2-to ratio. So it depends on the how many library is sequenced in the machine. So it's the random things, but only for the representation, I put the 2 and 2, right? Okay, so then you have multiple of this sequence information from the machine. So not just 4, it probably billions of that. Then you align that sequence information to the reference genome, right? So you align like in here, right? So this also takes the computational algorithm method. Then you can align this position, then you can read out T, right? So how many depths on here? 1, 2, 3, 4, 5, 6, 7. So 7 depths, right? So 7 reads you get from your DNA sample, then you cover this area. So you can say this area has the 7 depths. Then the coverage is this entire sequence, but about only 70% covered by reads. So you would say coverage would be around 70%. Okay, so this is the scenario 1. And the second scenario is the individual you used for your sequencing. So let's say this individual has the heterozygous allele, right? Homozygous allele for the alternative sequence, which is the different from reference sequence. So reference sequence is T, but your sample's genome has C and C, right? So this is again for all the information here. But only difference is ATC, GGGT, ACC, TTC here, right? C on here, C on here, right? Then once you align that, it only has the 7C, right? Then the third scenario is the heterozygous. So you get 1 strand from your father has the C, and mother from T, right? T from mother, right? So C and T. So how the things happen the same, but here has the T, T, C, T, C, 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 T, right? Some has the T, some has the C, some has the T, some has the T, right? So it has the certain proportion of C and T allele, right? So in theory, you have the 50% chance for your heterozygous sequencing. This is the critical information, right? So if you do the 10 depth sequencing, so that means that at least the 10 times sequence on this position, then in theory, you have 5T, 5C, or 5 for allele 1 and 5 for allele 2. That's in theory, right? But in a technique, it always happens as a expectation. So it can have 4 to 6, or 6 to 4, or 3 to 7, or 7 to 3. So in that position, in that point, you can you have to start think about probability. You have to think about statistics. How we can trust the data set you get from the DNA sequence information. We cover this one in data the sections. Okay, types of genome sequencing. Okay, so genome sequencing from NGS, the technology is the whole genome sequencing versus whole exome. Whole genome literally means that you read out all nucleotide from the genome, and exome is only read out the 1% of the genome. And microarray and NGS differences is the text SNPs or the entire readout we learned from previous, and that's all same things. Okay, so this is all good. And okay, so the review I used in here is from the 2016 the review. So yeah, so yeah, this is for your textbook for the session. I'm being posted out by, I mean, what is the direction of this? I think this direction is correct. I think the video is something incorrect. Yeah, I can check it. Okay, so you can read out this review. NGS technology, I emphasize again, this is, you know, I have no more words to emphasize, you know, this is the technology of these days. I mean, NGS is, you have to understand. I mean, everyone wants to use it, and this is like, you know, iPhone in or Apple machine in a biology science. So I mean, without knowing NGS technology, I mean, there's no way you can improve your science. So please read this review for your learning, and also keep in mind that how the NGS is worked out from the DNA samples to the data generation. And we can continue to discuss from the data modality of the next generation sequencing. Also highly recommend read out the presentation before the class. Okay, so everything is good, and see you on next Monday.